My name is Stephanie Myers. I'm a PhD graduate student at Oklahoma State University Center for Health Sciences. My presentation is called β-Funaltrexamine protects against lipopolysaccharide-induced neuroinflammation and behavioral impairment. One of the commonalities present in a multitude of neurological disorders is inflammation. For this reason, targeting inflammation has emerged as a viable option for the potential treatment of neurological disorders. Previous studies indicated that β-Funaltrexamine, a selective mu-opioid receptor antagonist, not only inhibited inflammatory signaling in vitro and human glial cells, but also inhibited LPS-induced neuroinflammation and sickness-like behavior in mill mice when administered immediately post-LPS. The present study explores the extent to which beta-FNA is protective when treatment occurs 10 hours after LPS administration and its effects at 24 hours after treatment, as well as the effects of beta-FNA on female mice, which to our knowledge has never been done before. For this study, we used male and female C57 black mice, an LPS dose of 0.83 milligrams per kilograms IP, and this is a well-established dose that causes inflammation and sickness-like behavior. The beta-FNA dose at 50 milligrams per kilograms IP given either immediately or 10 hours post-LPS, and this dose is based on a 2017 publication. We then conducted a 10-minute open field test to assess for sickness-like behavior, and here a normal mouse will walk around the box and explore, as you can see here, whereas a sick mouse will not, and they will actually stay closer to the walls and closer to the corners. We then conducted a 5-minute elevated plus maze test to assess for anxiety-like behavior. If the animal explores out into the opened arms, they are considered to have no or limited anxiety-like behavior, whereas if they stay within the enclosed arms, this is indicative of anxiety-like behavior. We then collected the whole brain, hippocampus, frontal cortex, cerebellum, brainstem, and plasma, and used an ELISA to assess cytokines and chemokines. Specifically, we looked at CXCL10, CCL2, IL-6, IL-1 beta, and TNF-alpha. Now, there were differential effects between whole brain versus brain regions, peripheral versus central effects, sex effects, and there were also variations depending on which inflammatory factor we looked at. But some of the most interesting findings were as followed. Now, our mice were broken up into 4 groups, our first group being our control in white, our LPS only in black, LPS plus beta-FNA immediately in teal, and LPS plus beta-FNA at 10 hours post-LPS shown in the gray. And what we're seeing here is that there is more sickness behavior in females based on the distance moved, and beta-FNA was not protective. We're also seeing that in the duration along the walls, there is no difference amongst any of the groups, this suggesting that there was no anxiety-like behavior going on. However, an elevated plus maze is a more accurate measure to assess anxiety-like behavior, which is why we did that next. And what we get to see is that at least within the frequency of the opened arms, the LPS groups are going down compared to the saline groups, suggesting that there is some level of anxiety-like behavior in both males and females. However, the decrease did not reach a level of significance. Interestingly, when looking at the cumulative duration in the open arms, the LPS group significantly spent less time in the open arms, indicating anxiety-like behavior in the males, and the beta-FNA treated animals were not significantly different from those of the control. And a similar trend was visible in the females, however, not to the level of significance. When assessing the effects of beta-FNA on LPS-induced CXCL10 expression in the plasma, we saw elevated levels in both males and females, and beta-FNA was protective when administered immediately after in the males and slightly protective at the 10-hour time point. However, it was not protective in females. The effects of beta-FNA on LPS-induced CXCL10 expression in the brain showed that at least within the whole brain, there was an increase in CXCL10 levels across the board, and beta-FNA was not protective regardless of sex. When assessing the hippocampus, you still have that increase in CXCL10 within the LPS groups, but now you also have beta-FNA protection in the males. However, this did not translate to females. In the frontal cortex, we are also seeing beta-FNA protection in the males regardless of the timing of the treatment, and we also see that LPS stimulation was much greater in the males than it was in the females. To conclude, there were differential effects between whole brain versus brain regions, peripheral versus central effects, inflammatory factors, sex effects, and when looking at the inflammatory factor CXCL10, we also can see that males showed more protection in the plasma, but not females. There was also no protection in the whole brain regardless of sex. In the hippocampus, there was some protection in the males, reminiscent of what we saw in the plasma, but not in the females. And the same can be said for the frontal cortex, with males having a more pronounced expression of LPS compared to females. Moving forward, we would like to further assess the temporal effects of beta-FNA on neuroimmune signaling, examine the effects of beta-FNA on LPS-induced inflammatory signaling in peripheral tissue, such as the spleen, liver, small intestine, and colon. I would like to thank Dr. Davis, Dr. Curtis, Kelly McCracken, and Daniel Buck, as well as OSU and OCAS for funding my research. Thank you.